Okay, so here we're going to talk about DNA recombination. And recombination is going to be important for diversity within our genomes, of course. So we're going to talk about homologous recombination, in which we have a holiday intermediate that's going to form, which basically means you have four regions coming together. We'll also talk about jumping genes, transposable elements, which are important in our prokaryotes. And we'll describe a translocation that is important in eukaryotes for us for diseases, especially in things like cancer, moving a oncogene into a region where now it can be expressed to high levels. So for recombination, there are going to be three main types. There's going to be homologous, and this is important for genetic exchange. And then there's also going to be transposons, which basically means you have elements in the genome that can move, and these are often called jumping genes. And then we have chromosome swapping or translocations, which can lead to diseases as well. So first, let's talk about homologous recombination, and this is a genetic exchange that occurs between molecules of DNA that share a region of homology, and so that's where it gets this homologous recombination. So it's genetic exchange. And so our chromatids, our sister chromatids, will line up in regions of homology, and they will swap information. And it occurs frequently when the sister chromatids are paired during meiosis. Now, this does not occur during mitosis, which is our somatic cells are dividing. This is our germline cells. And so in our homologous chromosome, we're going to have your chromatids that are going to line up. Let's see if I can show this. So you have your homologous DNA molecules, shown here, 1, 2, 3, and 4. And then you're going to have your chromosomes line up. You have a break in the chromosomes, an invasion by one of the chromosomes that crosses over. And then you have an exchange of material and a ligation. So now your two chromosomes have different genes associated with them. You're going to form what's called a holiday structure. And then the holiday structure is where you have these, basically you have the four chromosomes lining up. So it contains four double-stranded arms joined together, and the arms can adopt several conformations. And then you're going to have exchange of material, of course, after the exchange of material. So this is the holiday intermediate. And then you're going to have your release, cutting and resealing of the different chromosomes. And now you have your exchange of material on different chromosomes, for example. And so homologous recombination is a way to transfer genetic information. Now, for our replication, recombination can occur during prophase 1. And your DNA is going to involve the holiday intermediate. And it can also, and so that's usually where the genetic exchange of information is going to occur. And remember, in meiosis, we also are going to have our, has two cleavages, or your gametes are going to form through two separation steps. And it's in that first separation step where your chromosomes line up where you can get this homologous recombination. The next one is really straightforward and pretty simple. It's called a transposition. it's hard to believe that this happens, but we do have these in genomes where you have mobile genetic elements. And these mobile genetic elements or transposable elements can move from one region to another within a chromosome. And so these can move within a chromosome or to another chromosome. And there's no requirement for sequence homology. They simply just can insert themselves wherever they want to go. These are going to be important for our prokaryotes. So our prokaryotes are going to use these, such as bacteria. And they're going to use these to move around virulence genes. Antibiotic resistant genes. And one thing that our prokaryotes have that we don't have, or they have genetic elements called plasmids. And these are self-replicating genetic elements. So a lot of these transposons will move these types of genes within these plasmids that can then get passed from bacteria to bacteria. And you can quickly make a colony of bacterium resistant to an antibiotic, for example. And so here's an example of our transposons. So we're going to have our recipient DNA, and then we're going to have transposases, which are going to break the DNA open. And then you have a genetic element, transposon, that's going to jump into those breaks. And now you have incorporation of your transposon at a new location. And then finally, really fast, translocation. So translocations occur by breaks. So these are going to follow chromosome breaks. And these breaks can occur through UV radiation, sunlight, X-rays, carcinogens, and they can, all of these can result in gross chromosomal rearrangements. So you can, these are very large. Okay, and so in this case we're showing we have a normal chromosome 8 and we have a normal chromosome 14, and then we get a translocation where you're going to swap the ends of each of these. So if the free ends of different chromosomes are broken and then resealed together, you get a translocation, and this is going to be frequent in a particular lymphoma called Burkitt's lymphoma. Now, what's happening in Burkitt's lymphoma is that you have a proto-oncogene which is called c-Myc, and this is going to be translocated to the B cell antibody region. And so c-Myc basically moves from a low transcription to a very high transcription. And so the proto-oncogene gets expressed to abnormally high levels, and that can lead to cancer, a B-cell lymphoma. So we have rearrangement of sequences, and this is natural during our when we generate gametes, homologous recombination. Bacteria, they're going to use recombination as a repair mechanism. I didn't really talk about that, so don't worry too much about that. Eukaryotes, we're going to have recombination during mitosis. And then, of course, we have transposons, which are going to jump between chromosomes, within chromosomes. It's important for our bacteria. Transposons are important also for genes such as antibodies. And then we have translocations that are important for cancers.